Julia is from my team, so I'll give a little bit of introduction. Julia started working in my neuro lab during this summer, last summer. She's working on fMRlusic, which is a neuroimaging project. So there's a core neuroimaging based on analyzing fMRI data. She'll give you more details about what this project is all about. A little bit about Julia. Julia is a very passionate, hardworking researcher. So if she ever missed any meeting, she get back to me. First, she will let me know. It happened very few number of times, but she'll let me know. She'll ask how she can get to the work that she can, you know, make it up. She is always on deadline. She's very, I would say she's very smart, very intelligent, very hardworking, and she's like exemplary of being an awesome researcher. So here goes to Julia.
Thank you so much. All right, I'm gonna share my screen. Okay, can everyone see my screen? Great. So hi everyone, I'm Julia Wind, a junior at the Nueva School. I'm in Ms. Jahanikia's fMRlusic project, presenting on our research and the BIDS format. So today I'll be covering our computational data analysis project, including the basis of fMRI, the neuroimaging tools we'll be using, and our results thus far. I'll also be presenting on the BID structure and its impact on the neuroscience analysis community. So firstly, in our project at ASDRP, by using neuroimaging tools and techniques such as AFNI and FreeSurfer, we will pre-process and analyze an fMRI dataset obtained from the Psychoinformatics lab at the University of Magdeburg in Germany. The fMRI scans are 7 Tesla high resolution and also include a cardiac and respiratory signals. The dataset comes from fMRI scans of 20 participants who are shown clips of the movie Forrest Gump with different genres of music featured in the movie, such as country, symphonic, and 50s rock and roll. The participants were asked to guess the genre of the piece of music with and without audio. So the aim of our research is to identify the networks of the brain associated with guessing a music genre correctly without audio. In previous studies, researchers have gathered a large amount of data regarding the functions and growth of our auditory network. One key aspect of the network is its relation to music and its effects that music has on the brain. Both playing and listening to music have been found to increase the plasticity and strength of the brain. These activities also trigger reactions that are only achievable through musical stimulus. So we aim to understand and explain the role of the auditory network and genre association. Functional magnetic resonance imaging, known as fMRI, is a readily available, non-invasive neuroimaging technique that can be used to measure brain activity. Different signals and pulses produced within the magnetic field of an MRI machine are employed to observe the relative activity in brain areas from sensory-related tasks or stimuli. Of these signals is an MR signal, employed to indirectly detect levels of activity in specific brain regions using the blood oxygen level dependent or BOLD response. When neurons in the brain respond to a stimulus or task, they require oxygen from blood. Changes in MR signals result in blood oxygen levels heightening when there's an increase in brain activity as opposed to the resting state. BOLD signals provide the basis for fMRI and allow researchers to compare resting and stimulated states of the brain. The difference between MRI and fMRI is the images that they provide. fMRI provides the images of the brain's metabolic function, while MRI provides the anatomical structure. Unlike MRI, fMRI allows for real-time observations of changes in brain activity through the BOLD signals. An MRI can be used to scan for evidence of brain injury, stroke, etc., while an fMRI can be used to monitor the growth and function of diseases and tumors in the brain. There are many functionally separate networks of the brain that can be measured with fMRI. Networks of particular importance to fMRI scans are the resting state networks, as fMRI analysis requires comparison between the active and the inactive states of the brain. The most widely studied resting state network is the default mode network, which is active under resting conditions but inactive when the brain performs a task. Likely the most relevant network for the given task, however, is the auditory network. The auditory network is a collection of systems for hearing and decoding frequencies. In the ear, the cochlea converts vibrations into electrical signals, which are then sent up the auditory nerve. From there, the primary pathways signal the auditory cortex, while the secondary pathways signal reflexes. Located in the temporal lobe, the auditory cortex conducts high-level sound processing and perception. Each part of the primary auditory cortex corresponds to part of the cochlea in our inner ear. The auditory cortex is located bilaterally, and the right cortex has also been found to be more sensitive to tonality in music. The left cortex handles speech and language processing. We require different tools and platforms to pre-process, analyze, and run applications for fMRI data. For our research, we have decided to utilize fMRI Prep, an overarching fMRI data preprocessing pipeline that uses assorted tools from additional software, such as FreeSurfer, AFNI, and FSL. fMRI Prep follows a glass box philosophy, providing detailed visualization and documentation with each preprocessing step. Because fMRI Prep combines all the best neuroimaging tools into one platform, it is easy to use and makes it easier for reproducible results. FreeSurfer, an application fMRI Prep uses, is a collection of tools using structural and functional data to reconstruct the cortical surface of the brain. Among other things, FreeSurfer has the ability to transform the 3D volume of the brain surface into a 2D map. Another application, AFNI, is an open source environment for preprocessing, analyzing, and visualizing the 3 fMRI modalities: anatomical, functional, and diffusion-weighted. Batch processing using shell scripting is also possible using AFNI to automate the preprocessing of data. FSL is another neuroimaging and analysis software package with a library of data for analyzing these 3 modalities of fMRI data. Together, these applications are what we plan to use within fMRI Prep, along with Docker, an open platform for developing, shifting, and running these applications. Docker allows users to separate applications from their infrastructure to deliver software quickly and efficiently. The Docker containers are lightweight, portable, and executable, which allows us to isolate code into a single container to easily modify and update programs. The consistent environment of Docker containers implements reproducibility in our process. For our project, we selected our data from the Open Neuro website from research about music and auditory networks. To test the reproducibility of their preprocessing, we will convert the unstructured data to structured data ourselves. In this preprocessing, we will use fMRI Prep, bits conversion using Hue-Dye-Convert, and clean the data from MRI-associated noise signals. After these initial steps, we will analyze the data through machine learning and network analysis. We hope to find the relationship between different networks of the brain, particularly auditory, visual, and memory networks. We also aim to examine the role of the default mode network and accurately guessing the genre of a music clip without audio. So our team has successfully validated a mock dataset of the unstructured raw fMRI data for one subject into a structured BIDS format. We have recently received appropriate server space required for the large amounts of data, which is roughly 2 terabytes. We were also fortunate enough to study the state-of-the-art neuroimaging techniques currently employed in the field. By examining various neuroimaging tools and techniques, we determined that the best tool for our research is fMRI Prep. We have decided to additionally implement reproducibility in our analysis by learning and running our analysis on the Docker platform. So I briefly mentioned that we were going to be using Hue-Dye-Convert to convert our data to the BIDS format, but what is BIDS? BIDS was created by the International Neuroinformatics Coordinating Facility to efficiently organize neuroimaging data, and for our research, to organize fMRI data. To quote the BIDS website, neuroimaging experiments result in complicated data that can be arranged in many different ways. So far, there is no consensus on how to organize and share data obtained in neuroimaging experiments. Even 2 researchers working in the same lab can opt to arrange their data in a different way. With the brain imaging data structure, known as BIDS, it describes a simple and easy to adopt way of organizing neuroimaging and behavioral data. For example, in this diagram, you can see the unorganized data in the left in DICOM format, and the same data in BIDS format on the right. The right is organized and much easier for a human to understand. The BIDS format first requires all scans, the anatomical and functional, in DICOM format to be converted into NIfTY files, with the corresponding JSON files that include information about the scans. There's also a very particular naming system for the files to ensure that anyone who comes across the dataset will be able to understand what the file is. This can be done through the application of the microgl and the DCM2NIX command. They are then sorted into the appropriate ANAT and func folders for each subject of the study. A participants.tsv file and a data description JSON file are also required to list out all the participants and provide a general overview of the study. A dataset can finally be checked to see if it is in the valid BIDS format by uploading it to a BIDS validator. Labs using the BIDS format are able to share readable data between each other in an easily understandable format, knowing that not only will they be able to interpret each other's data, but their analysis tools will as well. Lack of consensus or a standard way to format files leads to misunderstanding and time wasted on rearranging data or rewriting the scripts expecting a certain structure. And because of the uniform file format that BIDS provides, tools and resources for neuroimaging analysis tools have flourished. Most neuroimaging tools now use files in the BIDS format as their input, which is why we are converting our data to BIDS before processing. These often open source programs that work on any data in the BIDS format allow researchers to build off each other's work in creating collaboration and reproducibility between labs, countries, and even time. So now that our data is on the ASDRP server, we have decided to convert it to BIDS format. We will use UDI-convert because for our purposes, it is the most flexible and has the most available documentation. We have begun the process of inspecting our data, looking for inconsistencies in tasks, quantities of runs, etc. For a simple example, part of our cleaning up process would be that if some tasks have 3 runs and some tasks have 8 runs, we would eliminate those extra 5 runs to create consistency across the subjects. After the data is preprocessed, meaning that it's cleaned up and in the BIDS format, we will use BIDS apps and tools mentioned earlier to analyze and visualize our data. Thank you so much. I'd like to acknowledge our amazing advisor, Sahar Jahanikia, and my team, Aditya, Anisha, Brandon, and Drew, who cannot have done this without. I'd also like to thank ASDRP and their funders who made all this possible for high school students. I'd also like to thank Sahar for her MRI scans that are used in this presentation. Our works cited is available at this QR code because it is way too long to fit into a slide. Are there any questions?
Well done.
I'll ask a few questions because I would lose face if I didn't. So you talked about functional MRI versus the classical MRI, and you said that the classical MRI measured structure, but this measured activity.
Mm-hmm. Yeah.
Is fMRI location specific? Can you get where in the structure the activity is occurring?
Mm-hmm. So MRI and fMRI are actually done on the same machine, just with slightly different settings. So both of them are looking at slices of the brain, which we later assemble into a 3D structure. But in fMRI, we're looking at, maybe I should pull up the slide. Okay, so in fMRI, we're looking at the increase of blood flow compared to the resting state. So we can see where the blood is moving, and we assume that the brain is being more active in those areas.
So you're looking for different resonances with fMRI and MRI, and you have to have different settings.
Mm-hmm.
Can these be done simultaneously in the same run, or do you have to do 2 runs?
I'm not 100% certain, but I think you have to do 2 runs, one for the structural scan and one for the functional scan.
So you don't actually do any scans, you just use data that's available.
Sorry, could you phrase the question again?
You don't actually do MRIs yourself. You just use MRI data that's available on the web.
Mm-hmm. So in this project, we're just analyzing data that we've found in online.
Do you have capabilities for doing your own MRIs at some point in the future?
At ASDRP, currently, we don't have an MRI machine.
Yeah, if ASDRP buys an MRI machine for Sahar, she will let all her researchers collect data.
Oh, okay, so time to talk to Edward.
Interesting.
So the hi,
Edward is just 2 million. I think it was 1.5 now. A 7 Tesla will be around like $2 million. It's just nothing, just one donation maybe.
okay, so I have another question. So the bids, the idea of getting it into the bid structure. So bids is kind of like JSON for brain image data.
I don't know what if you're referring to a different JSON, but the JSON files that I was talking about are the metadata files for the Nifty files, which are the 3D images of the brain.
Okay, and so I guess my question in that area is, does the neuroimaging data that's not include the structure also in JSON files?
I think all of the 3D files, whether they're structural or they're functional, have their own corresponding JSON files and other metadata files.
Okay. So when you say metadata files, it actually has the structure, but it also has the values of the readings in there too, right?
The structural and functional are different files. And so each of them has their own metadata files, which describes things like their runtime and other things like that. But all of the information about the structure and their function are in those main files.
Structural files would have location in the brain and some kind of mapping of where it is.
Mm-hmm. Yeah.
Okay. All right. Excellent. I will quit monopolizing the questioning and let it go to someone else.
Oh, hi, Julia. I thought you had a really interesting presentation. I kind of had a question about the study you mentioned that you kind of based your research on. I might have missed this, but if I remember correctly, it was about how testing like auditory sensors in the brain with videos without sound.
Mm-hmm. So part of the study was that the subjects would look at scenes from the movie Forrest Gump and try to guess what genre of music was playing in those scenes without actually hearing the music themselves.
I see.
Have these persons seen Forrest Gump before, or was this their first time seeing the movie?
I don't know the answer to that, but my guess is that these would be people that haven't seen Forrest Gump before.
Yes, so let me explain a little bit about the original study. So first they put them inside the scanner and then let them watch some video clips, and there's audio, and they ask them to guess the genre. And then for the second run, so it's just a normal test, it's just the control. And then for the second run, they turn off the audio and just ask them to guess the genre of a music without listening to the audio. Now, my assumption is that now that the auditory network is not working, so your visual network comes to picture, your memory comes to picture. So we have to look at the networks of how we can now look at what are the network associated to guess a correct genre of a music without even the auditory network comes to the picture. So we want to see what are those networks are associated with that. And
I have a word for you.
Go ahead.
Synesthesia.
Synesthesia.
Synesthesia is the production of a sensory image in one sense generated by a sensory input in another sense, like visual to audio, audio to taste, taste to smelling, something like that.
Correct. So then, so basically how you want to do that, and then you want to see that you want to predict also using machine learning algorithms. So when it comes to functional connectivity of the brain, after you analyze the brain, you just get the brain signal, right? So you want to see the correlation across the time, across all the slices of the brain that are highly correlated, and then guess, knowing one signal for a specific genre, we can guess at what time for example, ambient music was playing. So based on the correlation analysis and basically the time series of the brain after we analyze the brain and we get the time series. And then we have, in the behavioral scan, we have the associated runtime that shows that, at what specific time what task was playing. So we can correlate all this time-wise to basically do the correct guess of what is going on in that specific time and how brain detected specific genre without listening to that. So that's kind of a machine learning part of it that comes pretty later based on looking at the time series and the correlation between the time series. Okay, I think you have some questions.
I have a question, yes. So Julia, first of all, very rigorous treatment on the area of research that you're working on. It's very impressive. In chemistry, we have an instrument called nuclear magnetic resonance, which basically has a very similar principle. You use magnetic field to align NMR active nuclei, and then you look at resonances that are returned when an RF pulse is given. And in chemistry, a higher field magnet usually results in better sensitivity and better resolution. So I'm wondering if are there data sets from, say, different strengths of MRI magnets, and can you comment on whether that is a factor at play into how much resolution and how accurate your brain scans are?
It's been a while since I've studied fMRI. Sahar, do you?
Yes, so this is actually the study that they use the 7 Tesla scanner, the most high resolution scanner in the world that they, and it's also because like I personally work on so many 5 Tesla's data, and all the tools that has been developed so far is just with that low resolution brain. But this is actually, I decided to go with the 7 Tesla because it gives the high resolution, less noise. So the spatial resolution is much higher and better. But then it is, so once it's a rule with the scanner, that once you use a specific scanner, they have to stick to that one. So I don't know if this is what you were asking, but it is, yeah, I mean, the data is going to have less noise, hopefully, and it's of high resolution because of the 7 Tesla.
Okay, no, thank you. That's very helpful. So with a higher field MRI magnet, you have better resolution and more detail you can see, if I'm understanding that correctly.
Yeah, I mean, with 5 Tesla, we generally have an extra step in neuroimaging, which we do parcellation, we do a lot of resolution to just increase the, because the images are in 3D and 4D, they're voxel wise. So they're gray matter and white matter. So when it comes with low resolution, we have to intensify the gray matter so that we'll be able to see the activities, which is all the things has been done so far in field of neuroimaging by so many neuroimaging tools to just increase the resolution. But the good thing is that the data set that I selected for them is of high resolution, but the downside of it is that not many tools are actually developed these days to deal with the high resolution because you don't have to remove so many noise. And if you implement those methods, we might eliminate really some actual data as noise. So it's a little like, you know, it's it has its pros and cons, but I think going down the line, we want to move just with the 7 Tesla data. We'll figure it out. We'll figure it out. Okay, next question for you, Isaac.
Okay, so Isaac says that I thought that synesthesia is something that specific to certain people develops through specific experiences or genetics, meaning a constant connection, but this seems to be trying to measure the connection between ordinary people. So wouldn't this be a similar but unrelated phenomenon? Possibly unrelated, but no obvious distinct connection.
Not very familiar, me too. I mean, yeah. Julia, you have anything to add?
Okay. The way that I understand synesthesia is that it's a constant thing rather than trying, like actively trying to seek out the other sense based on the first sense. So I would guess that the brain activities are slightly different.
Yeah, I mean, it looks like there are specific people, as I'm understanding from that. First of all, I've never, as a researcher in this domain, never come across this issue. But what I'm understanding that there are people with ability that they can, you know. Okay. Please mute. Kirtana, right? Am I pronouncing your name correctly? Thank you. Yes. So I'm assuming there's some people that they have ability to use one sense instead of the other one. But in my research, I've never come across that. So I don't want to pass comment on this. And it's totally irrelevant with what we're doing because it's not looking at the people abilities, but I think what comes to the picture is mostly like, I think after we do analysis, we're going to see a lot of activation in memory, because what happened, like and visual network, and there should be a connectivity in the brain between these two regions, obviously. But what do you think, like, when you see a rock band, you see like people are screaming, you see a guitar, you see the drum. So like, of course, like you can see, you can use your memory, what you have in your network to just recall what kind of genre is that. And of course, your visual network comes to picture. But these are obvious. But what I was concerned is to look at the default mode network, which I think will have an impact, or we'll be able to see some network associated with default mode network in specific study, which I'm very curious. It's just like my assumption, my hypothesis. It might even not come true, but I'm very curious and I'm emphasizing on this. And I would like my researchers to work on that one. Because we never know. Next question for you from Sahas, I guess.
Are additional psychological factors such as priming bias considered in our analysis?
Can you tell us what is priming bias means?
I'm just guessing, but I think that means previous exposure. So I think that would be something that would be considered when they were collecting the data rather than us analyzing their data. I think we're certainly going to look into, okay, so it is previous exposure. Yeah, so I don't think we're going to implement that in our computational analysis, but but I think we'll keep that in mind, but it's not going to influence our computational analysis.
I have a question from, I'm moving upward. So I have a question from Shashank. He's asking, how does use of beats add and subtract from overall system load when pre-processing this data set? Do you have answer for it, Julia?
Well, I know that converting to bids obviously takes more computational power, but I'm not sure. I haven't looked into structures besides bids that were optimizing for things like computational load or time. But in general, I think the main benefit of bids is for making those neuroimaging analysis tools and creating a standard input for them. But maybe someone can come along and create another standard where it is optimized for computational or system load. But I don't think that's what bids is optimized for.
Well, I mean, so it says, how does use of bids add and subtract from overall system load? It's actually, so first you have unstructured data, right? So before that, every lab in every part of the world, they use their own data and then put it one by one or through a code. Generally, they use MATLAB neuroimaging or neuroimaging researchers, or they use shell scripting. They put it inside the pipeline. Now, what happened? It was time consuming, and then the result, like literally from one lab in the first floor, from the second lab in the second floor of the same building, was like totally different when they do the same tools. So that's why bids came to the picture. So I thought, okay, let's have the unstructured data into the structured data. And now let's create a pre-processing pipeline that just accepts that bids format as an input. So it does not add or subtract the load. It's an extra step. First, you make your data in an unstructured way. Now you pass it to any neuroimaging pipeline, you should get the same result out. And that's a use of that bids. From the load, computational load, it's definitely reducing some time, a lot of time, actually, not some time, a lot of time, especially for the researchers that they don't have computational background and they do manually one by one. I have seen it in my eyes. Trust me, it's real. Like a year of just putting one by one every file to just looking at the neuroimaging instead of using bids or automating the system. But it's not side by side. It's done, and then the output goes through as an input of the neuroimaging tools. Okay. I think that's all. Anyone else? I think you have a new question.
For the study you mentioned, are factors like the age of participants or how often the participants listen to music counted for in the data? Will such factors even influence results? As for the last question, I'm sure that's a topic of a different research study, but I'll have to go back and look at the study. If you put your email in, I can get you an answer to that later.
So I'll add, the study is about young adults. So when it comes to neuroimaging, sorry to say that, but 40 plus is considered as an old brain. So when we say young adults, that means the data is collected between age 18 years old to 40 to maximum 45. Trust me, I never went even more than 43 because I don't want to deal with the brain kinds of, you can see the aging in most of the brain. So you want to avoid that because that affects your data. It is not a reality. That doesn't mean that I'm saying that 45 plus is an aged brain, but it's a higher chance being towards aging. So you don't want to risk collecting data and then eliminate them. So this, you go safe and you collect 18 to 40 or 45 max. I would say 40 still. So these are young adults. That means you're talking about 18 to 40. So how often? No, it's just anyone. They're just like healthy participants. So when it comes to fMRI, unless it is, unless it is very specific to any psychological background, will not look at that. So just like any healthy participant, come and look at the genre. Next question for you.
Would there be variations in the default mode activation with different genres listened to? I think that's one of the questions that we're trying to answer, looking at the activity during rest or when just listening to audio and trying to guess the genre from their activity. So we'll know in a year or two.
Yes.
Would someone's preference to a certain genre of music make any difference in how quickly or easily they would be able to guess what genre it is, both for audio and no audio part? I'm not sure for the audio part, just from observing people in everyday, not based on like neuroscience, I would guess yes. If you've been exposed to a genre a lot, you would be able to identify it quicker, I'm guessing. For no audio part, I'm leaning, again, not based in any scientific thing, but I'm leaning towards no. I don't know. What do you think, Sahar?
Well, I mean, yeah, it can be yes or no. You know, for entire your life, you have been going to classic. I mean, it should be very specific. I'm assuming a person who has never been exposed to any specific genre then can go wrong because they cannot detect. I mean, it's very common sense, like when you see heavy metal versus a huge symphony playing. So I would not say that it make a huge difference. It, of course, make it so. The difference comes when you have a recording of response time. So when you are inside the scanner, they'll give you 20 seconds so they give you, so you cannot talk inside the scanner because it's extremely loud, okay? So they give you a tool, which looks like the Xbox, the handle, the player. What do you call that, Julia? Xbox
button.
yeah, it has something, someone comes with the words, whatever, but the one that you hold and play. So it gives you in your one hand. So it's either have like one button or two buttons, or yes, thank you, the controller. Yes, thank you, yes. So they give you a controller, something like that, but it has button on it. So for example, in this case, it has 5 buttons. So they said that there's an instruction on the screen that the participant laid on inside the screen. So they see a very small screen, which is a reflection of the back TV. Okay, so there's a huge TV in the back side of the scanner, and through the reflection of 2 mirrors, they see in a very like small kind of, like literally this much is the size of the screen that they see. So there's an instruction for them that they have to read it very quickly. The instruction says that if you see if the genre of the music is ambient, press 1, finger number 1, 2 is country, 3 is heavy metal, 4 is rock and roll, 5th is symphony. So you have 20 seconds to answer to this, to select the genre of the music. So I'm assuming with this one, when it comes to the response time, a person who has been exposed and listening to specific kind of genre, or maybe to all kind of music, so the response time is very quick, right? We have sometimes that we don't record anything because participant was so busy ruminating over what is it, that 20-second time is passed. So that's why the response time for them, the response that they make is longer. So I would say that yes, looking at the data, we look at the response time, it's very important, but sometimes so response time, accuracy, these are the very important factor that after the scan is done, we look at it. But yeah, it is going to have that factor that you can see the brain responded quickly to detect that. But I don't think so it has much of difference in the network of the brain. I think we're done. Yes, because I think if they put questions in the question box, and me and you are going to go till tomorrow answering the questions. I'm very, very happy, very excited, and I'm very thankful to ASDRP to give us this chance to work on neuroimaging project. It's very exciting, extremely exciting. I love working on MRI data. I work with EEG data. It's well, signal analysis is not something that it bores me. I enjoy looking at the scan of the brain, but I'm so happy that my fMRlusic team is exposed to get the chance of looking at MRI data. I don't know. I hope they enjoy it and they get to publish a nice paper in a neuroimaging journal. You never know. But so that's from us tonight. If